Okay, so first and foremost, guys, I did not get through in my last 2 classes enough that I had planned for. So I was going to have your test on Wednesday. Your test on respiration is now going to be on Thursday because I just couldn't get through everything today that I wanted to get through. Hi, Aiden. Tell your kitty I said hi. We see him almost pretty much every class. I love it. All right. So first of all, I need you to watch lecture 4. That's your official homework assignment. I need you to grab your notes because I'm going to cross through a few pages and I want you to fast forward through a couple pages. So I need you to find page 26. That should be the start of lecture 4. If you watch the first 3 lectures, then basically you're going to turn until where your notes stop, which is page 26. It says lab measuring and determining respiration rate. Okay. This lecture is about a lab that there's no way I can perform. Like, you guys have to do this lab to really understand the parts to it. And I'm going to try to do that in the ecology unit when I'm talking about photosynthesis and respiration again in relation to ecology and organisms consuming oxygen and producing oxygen. Okay. So this lab is about measuring how fast you do respiration. Now, honestly, guys, I could measure your respiration rate. This is what I would do. I would lock you in an airtight chamber. And while you're in that airtight chamber, you would be consuming oxygen, right? So my oxygen levels would go down. And all I have to do is calculate your respiration rate is figure out how much oxygen I start with. Then after a period of time, how much oxygen you have consumed. And I can calculate rate, the change in oxygen over time. That's your respiration rate, okay? All right, is everyone on page 26? This is the start of lecture 4. You listen and fill out this page. Do not cross through page 26, okay? It gives you the background to this lab and how we can set up these devices, which are called respirometers. Those are like the airtight chambers that if I put a living organism inside of them, I can measure how much oxygen they're consuming to do respiration and to make their ATP, okay? Turn to the next page, though. This is page 27. I want you to fast forward through it. Don't necessarily cross through it. We might have to come back to it in May, but you can say skip this page. This is more about actually setting up the lab, and we're not doing that right now, so I don't want you to bother with the setup and the problems that we might have. I also want you to skip this next page, page 28. So you can write skip at the top of it, but since we're calculating reaction rate, you guys have done that before, you should be able to look at a graph, and if there's a change in something over a change in time, I should be able to calculate reaction rate by just the slope of the line, okay? So here's what we're going to do, and this is the only thing that we're going to write on this page, okay, is I'm going to calculate the rate of respiration of pea seeds that are germinating. So those are living cells. They need to make ATP, so they're going to be sucking in oxygen so they can make ATP because their cells are going to need it for cell division, okay? So germinating peas, it's this green line. So at the start, it looks like I started with 0.95 milliliters, okay? And that was at time 0. Okay, so 0 minutes. Remember how you do rate. Change in something over my change in time. Okay. And now I'm basically just going to draw. Now, if I say calculate the average rate, we'll just do the beginning and the end data point. So my end time is 30. So minus 30. Hopefully this all rings a bell, right? To calculate rate, change in something over my change in time. And at 30 minutes, it was 0.61. So minus 0.61 milliliters. And then I calculate rate. Okay. My units, when I'm done, I'm going to have, I don't know, I didn't calculate it, but it's going to be a number and it's milliliters per minute. Do I times by 100? No, that's percent. This is rate. Okay. Something different. Okay. So just make sure you can look at a graph or look at a table and calculate the change in something over time. And that gives us a rate of something. In this case, it's going to be respiration rate. How fast you're taking in oxygen to make ATP. Okay. Now, using your finger, up or down. What do you think is going to happen to respiration rate when you're exercising? All right. So the more active you are, the higher the respiration rate. Okay. One other thing, when you're cold, when you're outside, let's say it's 10 degrees and you have a light jacket on and you're cold, do you think respiration rate is going to go up or go down? Nice. Okay. So when you're cold, you guys all know that you shiver, right? Shivering is the best workout. If you shiver for 10 minutes, that's an hour's worth of moderate exercise. So you need to rethink about shivering is just working out. And you just told me if you work out, your respiration rate goes down. If you are in a warmer or a cold environment, you start to work out, you start to shiver. And so you actually start, your breathing will start to increase because you have to make a lot of ATP to flex your muscles because you're cold and your brain is trying to get you to generate some heat. Okay. All right. We'll talk more about that here in just a little bit. All right. So we need to quickly review because it's been too many weeks and we need to go back and just look at what we've learned about so far. Okay. That's going to be kind of the next 20 minutes and then we're going to go through new stuff. So I need you to grab your posters and I need you to turn to the side of aerobic respiration. Now, if I say aerobic respiration is taking place, that means that 4 steps are occurring, 4 metabolic processes are occurring. Glycolysis, the prep reaction, Krebs cycle, and oxidative phosphorylation. Okay, that's the 4 steps of aerobic respiration. Okay, so in a second, you guys are going to go do a problem that usually takes students a long time to do. But if I go through your posters with you one time, then you guys can usually answer this question very, very quickly. And you're going to do that in your breakout rooms. Okay. All right. So first of all, everyone find glycolysis on your poster. I'm not going to show my poster. You have to look at yours because you guys have to learn to read it, to study it, to do well for your test. Okay. Okay, hopefully this makes sense. Follow along. Glycolysis takes place in the cytoplasm of cells. During glycolysis, glucose is broken down, or you can say glucose is oxidized. To start this process, the cell has to use 2 ATP. As glucose is broken down or oxidized, protons and electrons are removed and placed on coenzymes such as NAD+. NAD+ is getting reduced. The intermediates are getting oxidized. When NAD+ is reduced, it forms a molecule which is called NADH. NADH is a proton-electron carrier. It can carry protons and electrons to the electron transport chain in the mitochondria. Okay? So in glycolysis, we get 4 molecules of NADH, 4 molecules of ATP, which is a net of 2 though, and you get 2 molecules of pyruvate. Okay, next step. The pyruvate can be sent into the mitochondria where the prep reactions take place. Pyruvates get broken down. You can say they get oxidized. Protons and electrons are removed and replaced on an NAD plus to make NADH. Also, a carbon atom gets removed. And now we have a molecule which only has 2 carbon atoms, which is called acetylcoenzyme A. Okay. Step 3, the acetylcoenzyme A is processed in a series of reactions. The breakdown or the oxidizing of acetylcoenzyme A produces 3 molecules of NADH. The 2 carbon atoms are removed as CO2. We get 1 ATP produced, and we get another coenzyme being produced, which is called FADH2. Now, glucose has been completely broken down. 6 carbon atoms have been removed, 12 protons and electrons have been removed, and same with the 6 oxygens. Now, for the 4th step, it's called oxidative phosphorylation. It takes place at the electron transport chain proteins, which are proteins embedded in the inner membrane of the mitochondria. NADH and FADH2 are oxidized at the electron transport chain. They release their protons and electrons and turn back into NAD+ and FAD+. Watch the electrons. The electrons from these shuttles are passed down from protein to protein. They're passed down the electron transport chain, And as they are moved, H pluses are moved out of the matrix and into the inner membrane space of the mitochondria, where they become concentrated. These protons can move from high concentrations to low concentrations. They move through a molecule called ATP synthase, which provides energy for ATP synthase to phosphorylate ADP, which means it takes ADP and it adds a phosphate group to make ATP. Now, protons can be concentrated in the inner membrane space only if the electrons can move down the electron transport chain proteins. Oxygen allows for the continuous movement of electrons. Oxygen is known as the final electron acceptor. It can remove the electrons from the electron transport chain proteins, so the electrons can continue to flow. In that process, it also reacts with those H pluses, and that forms water. Okay, so that's where oxygen is needed in respiration, and that's where water is produced. Okay, all right. Now, real quick, go ahead and flip over to the other diagram that shows you anaerobic respiration. Only 2 steps happen in cells. In an animal cell, glycolysis happens because it doesn't need oxygen. Glycolysis is considered an anaerobic process. Aerobic means oxygen. When you take aerobics, they don't want to kill you. They want you to breathe all throughout and get plenty of oxygen. That's why we name that exercise aerobics. They want you to monitor your breath. Anaerobic means no oxygen. So glycolysis can happen when a cell isn't getting oxygen. Notice, does glycolysis look any different on this diagram compared to the one we just covered? It's the same step. Glycolysis happens, okay? But if there's no oxygen, then the NADHs and the FADH2s, they cannot be oxidized with the electron transport chain. So the electron transport chain shuts down. You cannot unload or oxidize those coenzymes. But we do have a molecule we can use. Glycolysis makes pyruvate, right? See the pyruvates? In the second step of anaerobic respiration, it's called the fermentation step. Pyruvate gets reduced. The NADHs can unload there, not at the electron transport chain, but it can unload protons and electrons onto the pyruvate. So NADH is getting oxidized, pyruvate's getting reduced. That reforms the NAD plus needed for glycolysis. So on that poster, do you guys see where it says regenerates NAD plus? That is the most important thing for you to remember about fermentation. The fermentation step frees up NAD pluses because without those, glycolysis would even stop. And glycolysis, show me, how many ATP do you get from glycolysis? So as long as glycolysis happens, at least you get 4 when you're out of oxygen. Okay, it's not very much, but at least it's some to keep you alive a little bit longer. Okay, now let's look. In lactic acid fermentation, when pyruvate gets reduced, it becomes lactic acid. Do you make any CO2? Using your fingers, how many carbon atoms are in pyruvate? Look on your posters. 3. Look at lactic acid. How many carbon atoms are in lactic acid? Did you make any CO2? Did you pull off any carbon atoms? No. So in lactic acid fermentation, no CO2 is produced by cells. Okay. Now, yeast cells, they don't do lactic acid fermentation. They do alcohol fermentation. So look at that diagram with the yeast cells. Glycolysis happens. Okay. But the cell's out of oxygen. So in this case, the pyruvates also are going to be reduced. The NADHs are going to be oxidized. And the pyruvate gets turned into ethyl alcohol. How many carbon atoms are in ethyl alcohol? Or the alcohol that I labeled. I didn't label it ethyl alcohol, but it is called ethyl alcohol. You see there's 2 carbon atoms? How many carbon atoms are in pyruvate? 3. So did CO2 get produced by the yeast cells? Yes, so they do produce CO2. That's why beer is bubbly. That is why champagne is bubbly, okay, because of the CO2 is being produced along with the alcohol. So it's bubbly, okay? All right, but then again, the number one thing is that the fermentation step provides NAD pluses for glycolysis, because without those, glycolysis would stop. All right, everyone grab that worksheet 4.1. It doesn't say 4.1, but it does say entropy and Gibbs free energy. And I want you to turn and take a look at question. Wait, I lied. I'm sorry, I lied. 4.3. Grab 4.3. And you're going to turn to question number 6. Question number 6 is about this diagram. A, B, C, H, etc. Those are molecules. So what you guys are going to do in your breakout rooms is you are going to try to figure out what molecule is A, what molecule is B, what molecule is C, and so on. And you're going to fill in, really just focus on the short line of A, B, C, and D. If you guys get done and you figure out all of A through H, what those molecules are, then you can go back and you can start to talk about the relation part of question number 6. Okay. All right. Give me just a second here. I'm going to open up 5 breakout rooms. I don't care which one you go to be honest. But I just need you to get in there and see if you guys can work together to figure out those molecules in 6. Stop. All right, I'm working on, there we go. All right. Okay, so a review of lecture 1. First of all, energy can never be created or destroyed. It drives me nuts on essay questions when you said energy is made. It's not made. It only changes form from one form of the energy to another form of an energy. Okay, so there's different forms of energy: mechanical, chemical, potential. There's light energy. Okay, so hopefully you can take a look at these 2 equations and tell me what process is this top equation.
Photosynthesis.
Okay, so in relation to energy changing forms, what plants can do is they can take light energy and they can turn it into glucose. Now, glucose is a molecule that has energy stored in it. So we say that glucose is chemical energy. It's a chemical, it's a molecule and has energy in it. Okay, down here we have respiration. Know your equations, have them memorized. In respiration, chemical energy, glucose, is used or converted to a different form of chemical energy, which is ATP. Okay, so energy can change forms. Now, when we change forms, energy changes from one form to the next. You always lose some energy. So for example, this is my daughter, Brooklyn. She's going to die someday whenever she is listening to this lecture and she sees her picture that I've been showing to students from when she was like in the 4th grade. So she is at Science City and she is burning chemical energy. She's on this bike. Okay, so let's say that she breaks down and she uses 100 kilojoules of energy to move her muscles. When you actually measure the amount of energy that goes to lighting up this light bulb, it maybe is only about 30 kilojoules of energy. 70 kilojoules of energy was lost as the energy was changed from chemical energy to mechanical energy to light energy. Okay, so energy can change forms, but no conversion of energy is ever 100% efficient. Okay, so let's talk about efficiency. So, Emma, I have a question for you. You took a test. Let's say you took a test. There was 100 questions, and you got 35 questions right. How do you think you did on that test?
Well, it obviously depends on how many questions there were.
There was 100. There's 100, and you got
100?
35 right.
So you did bad.
Okay, all right. So you weren't very efficient, right? You didn't learn very efficiently, okay? So just so you know, when we take in respiration, we're taking chemical energy and turning it into a different form of chemical energy. is we do lose some energy as heat. And that's one reason why we can stay warm, is because that heat can be used to heat our bodies, okay? So just kind of look at these numbers. You don't have to have them memorized. Just again, we're just relating ideas to respiration. Is that if we have 686 kilocalories of energy in glucose, when we're done with that glucose molecule, we can make about 34 ATP from that one molecule. When you take a look at the energy that's stored in 34 ATP, it's only 236. So what the heck? Look at all that energy that we lose when we release energy from glucose, okay? So this is just showing you that energy is lost when energy is changed from one form to another form. And that's what happens. No energy conversion is 100% efficient, right? Now, we're going to talk about some words here from lecture 1. Whether reactions are spontaneous or not spontaneous. Do these reactions increase or decrease entropy? Is this reaction showing a positive or a negative change in free energy? Is this reaction an endergonic or an exergonic reaction? Okay, so I'm going to take all these terms and I'm going to relate it to 2 reactions that we are focusing on this unit: photosynthesis and respiration. Okay, I'm also going to relate this to Legos, because I honestly think that how many of you guys have built Legos before? Seriously? Okay, I feel like everyone would have built Legos. Okay, so with Legos, you have pieces, right? And it takes you hours, usually, depending on what you get here, to turn those Legos into a highly organized structure, okay? It takes energy to do that. You guys have to input energy. If you dump the Legos out on your living room floor, they will not spontaneously turn into a castle, okay? So this is like photosynthesis. The pieces are the CO2 in the water. Notice there's 6 CO2s, there's 6 H2Os, there's 12 pieces. And when photosynthesis is complete, you have one piece. You have one glucose molecule that has those 6 carbons, those 12 hydrogens, and those 6 oxygens that we get here, okay? All right, so we organize these atoms into a more complex structure. So this is CO2 and water. The castle is glucose. It's an organized molecule. Remember, I had the model of glucose built. And I was like, it took me a good, 40 minutes to build this with these, the carbon and the hydrogen and the oxygen, all right? So let's relate photosynthesis to these Legos and these words up here, okay? So do you think CO2 and water can spontaneously come together to form glucose? No, there's CO2 around you right now, right? Isn't there also water in the air? Are you guys getting sugar made in the air around you and it's sprinkling down onto your desktop? No. So photosynthesis is a non-spontaneous reaction, okay? What word, according to the lecture, should I substitute in for entropy? What does entropy mean? What word could I use for entropy?
Order or chaos?
It's disorder. Disorder. Okay, so everyone take a look at the room that you're in. Do you think your room is highly disordered or has low disorder? Okay, so if it has high disorder, it's messy, right? Mine's pretty messy right now. My room has high disorder. I have stuff all over the place. Or I could say my room shows high entropy, high disorder, okay? All right, so let's look at photosynthesis. To go from this pile to the castle, did you increase or decrease disorder? Did you organize them?
Decrease or
Yes.
increase disorder.
Yes. Notice you made them neat. You took these pieces and you made them neat into one molecule. You decreased disorder. You organized them. Okay. So I would say photosynthesis decreases entropy. It decreases disorder. Let's take a look at respiration. Respiration takes glucose, here's my castle, and breaks it back down into the atoms that it's made out of, CO2 and water. So respiration increases disorder. That process increases entropy. Does it make sense? Okay, let's talk about the change in free energy. Actually, first I'm going to talk about endergonic and exergonic. If I have a prefix in, that means put something in. If I have a prefix X, like exit, that means take something out. So to go from the pile of Legos to the castle, do you have to put in energy? Yes. To decrease disorder, to make things organized, you have to put in energy. Like to clean a room, to decrease disorder, you have to put in energy. So if you put in energy to a reaction, it's called an endergonic reaction. Okay? Now, if I say, summarize, why do you do respiration? I think most of you would be able to agree with me. You would probably say something like, we break down glucose. We release energy from glucose and we use that energy to make ATP. Okay? So respiration releases the energy that's stored in the bonds of glucose. So respiration is an exergonic reaction. We get energy out of glucose and then we can use that energy to make ATP. Okay? All right. Now let's talk about change in free energy, whether there's a positive or a negative change in free energy. Now, a few abbreviations that you guys need to note here is that G means free energy. A triangle in front means change. Delta means change. So my change in free energy. So can anyone tell me, when you're doing math, what does the plus mean? What does a plus mean to do?
Add.
Thank you. Remember that. Okay, apply what you guys already know. You guys knew in the second grade or first grade even. Plus means to add. To go from this pile to the castle, did we have to use energy to make that happen? We had to add energy. So photosynthesis has a positive. You have to add energy. That energy is light energy, right? So photosynthesis has that reaction has a positive delta G. When you break down organized molecules into pieces, you release the energy. So my change in energy is negative. You have a negative change in free energy. Okay, so photosynthesis, you have to add energy. They use light energy, is what they use for energy. Positive delta G. Anytime you break down a molecule, you're going to have a negative delta G, a negative change in free energy. Energy is going to come out. Okay, now what that looks like on these graphs. Now, you guys have seen graphs like this before because I talked about it when I talked about enzymes. And you are going to see that we have, as time goes on, a change in energy of these molecules. Okay, so I'm diagramming the change in energy as a reaction takes place. So when we start with a reaction, we're going to start with the reactants. Okay, and CO2 and water, if you actually add up the amount of energy that's in them, it's pretty low. Okay, and we have to add energy. So if my energy starts low, when I'm done, when I add energy, I'm going to have a product that has more energy in it than the reactants because we added this much energy. Add is a plus. So this reaction has a positive delta G. We have to add energy to make it take place. Okay, so if we have to add energy, or if a reaction has a positive delta G, this reaction is non-spontaneous. You have to add energy for the reactants to turn into the products. And that's what happens in photosynthesis. Okay, all right. Now, when we look at respiration, respiration is going to take a molecule with a lot of energy in it, and it's going to break down that molecule, and it's going to release that energy. So we see we have respiration. We take a molecule of glucose. It's the reactant. It has a lot of energy in it. And we can break it down to the products. CO2 and water are the products when you break down glucose. And they have less energy in them. So from here to here, this energy is released. All this energy will come out of glucose, and we can use that energy to make ATP. So this is what it looks like when we have a reaction that releases energy and we say that it has a negative delta G. You don't have to add energy for this reaction to take place. This reaction is a spontaneous reaction. Entropy is going to be increased because we're going to go from orderly to disorder. Molecules will break down on their own over time. Okay, now your turn. I need you to go to joinpd.com. So open up a new tab and go to joinpd.com. We're going to see what you guys learned. Joinpd.com. It might take a second because, again, I got a lot of stuff up and so it's lagging a little bit. But if you go to joinpd.com, then you'll be ready to punch in this code when we get one here. Oh, shoot. Never mind. Hold on. I hit the wrong thing. Still go to joinpd.com now. Okay. While this is launching here, I'm going to show you the question I'm about ready to ask you. Okay. Actually, I should be able. Just wait. All right, there you go. There's your access code. Go ahead and jump in for me, please. SYIACC. Awesome. SYIACC. All right. All right, let's go ahead and get to the question I'm going to ask you. All right, here we go. So I have a reaction. Here I have a bunch of amino acids, and you guys have to put them together, remember, form peptide bonds to build a protein, to build a polypeptide. Okay, so that's a reaction. Here's our reactants, here's our products. You guys should have 3 dots. Take your blue dot. If this reaction can happen on its own, if it's spontaneous, put your blue dot on the word spontaneous. If this does not happen on its own, then it's not spontaneous. Put your blue dot on the not. Think of Legos, okay? And think of, are we building something or are we breaking down something? Are you building Legos or are you breaking down Legos, okay? All right, second thing, grab your yellow dot. You're either gonna put your yellow dot on an exergonic reaction or endergonic reaction. If to go from here to here, you have to add energy for this to take place, it's endergonic. If when you make a protein, if that releases energy, then it would be an exergonic. So go ahead and put your yellow dot. And then the last one kind of relates to exergonic and endergonic. If you have to add energy to go from this to this, then it has a positive delta G. If it doesn't take energy to go from this to this, then you have a negative delta G. All right, let's see how you all did. I bet it's half and half. Oh, look at you. You guys are doing well. You don't know how happy this class just made me because the other 2 classes just botched the heck out of this, okay? All right, so let's think about this. Let's think of the Legos, really? These are the Lego pieces and you put them together, like that's gonna take energy, okay? This does not happen on its own, okay? So this is a non-spontaneous reaction. So to get this to happen, we have to add energy. So this is going to be an endergonic reaction. We have to put in energy to take these pieces and organize them into a chain. And since we have to add energy, it has a positive delta G. Awesome. All right, let's go ahead and go to 4.1 and let's do question number 4 together. Okay, question number 4. And you can see that we are looking at a graph kind of to what we've seen before. We have a reaction happening. So that means that we start with the reactants. And when we're done, we're going to end up with products. Okay, so we got A done. All right, my change in free energy is how much we have to add from the beginning to the very end. So my change in free energy is just this part right here, from what we start to what we end up with, okay? And notice we're gonna have to add energy. So our change in free energy says calculate the change in free energy for this reaction is just the difference between here and here. We have to add 80 kilojoules, and that's gonna be my change in free energy. We start with 80, and when we're done, we have 160 worth of energy in a molecule, okay? Since we had to add energy to go up, because we went from 80 to 160, then we have to add, so we'd have a positive change in free energy. That's what delta G means, is change in free energy. Okay, and since we had to add energy, this reaction, what we're looking at here, we had to add energy. Ender means in, would be an endergonic reaction. And anytime you have to add energy, it means that these things don't happen on their own. This reaction will not happen on its own. So this reaction is not spontaneous. You have to add energy. That's like Legos turning into a castle. Not spontaneous. You have to provide the energy for that to take place. Okay, again, entropy. Just substitute disorder. Okay, if you add energy to your room, you are decreasing disorder. If we add energy to reactants, we're organizing them. We are decreasing disorder. So in this reaction, we're decreasing disorder. By the way, this is almost a classic graph for like photosynthesis. Start out with pieces, CO2 and water. And when you're done, you have a highly organized molecule where those pieces are built into a nice structure, like a castle. Okay. Calculate the activation energy. Okay, we've talked about activation energy before, and let's go ahead and do F, because we talked about graphs like this with enzymes, and we said that the job of enzymes is not to provide energy, but to actually lower the amount of energy needed. It lowers the hill. So this is your answer to number F. It says add a line to show the change in activation energy if this reaction is assisted by an enzyme. So enzymes lower the amount of energy needed to go from reactants to products, okay? Now, if I say calculate activation energy, and we can see that this just lowered activation energy, the activation energy is the hill. So without the enzyme, if we start here and we have to get up to here, that's the hill. This is activation energy, or energy of activation, okay? So how much energy did I have to add to this 80 to get to 240? It would be 160 kilojoules. Okay, 160 kilojoules. Okay, I want you to turn to 10. And turn to 10. And I'm just going to draw, there's 2 looks to this graph. The one we just did started low and ended up high. Okay, this is a graph that represents photosynthesis, when you have to add energy to make an orderly molecule. You're going to build something. When you see a graph that starts high and ends low, then that means that you are breaking down a molecule. You're breaking down the castle. Okay, the castle has a lot of energy. Glucose has a lot of energy. You're going to break it down. So this is like if we started with glucose We're doing respiration. And then when we're left with pieces. We have CO2s everywhere. We have water molecules everywhere. Our glucose is gone. Okay? All right. To go from 150 to 50, we don't add energy. We actually lose energy. So my change in energy is from the beginning to the end. So my change is 100. And since we're not adding energy, it's a negative 100. So my change in free energy is 100 kilojoules. It decreases. Energy decreases. Okay? From there, I want you to see if you can answer the other questions. Just take a couple minutes to see if you can answer B through G. Okay? So let's think about this. This is like respiration. We break down glucose and release energy from it. It's an exergonic reaction. Okay? Whenever we have a negative delta G, that reaction is usually more a spontaneous reaction. Things break down over time. Things break down over time. Think of your house. If you left your house and you came back in a million years, do you think your house would be there? No. Things break down over time on their own. Okay? All right. We have a negative change in free energy because we didn't have to add energy. Did we increase or decrease disorder? We went from one thing to many. That's the castle to the pieces. So we increased disorder. We made it more messy. We increased entropy. Activation energy is just the hill, guys. That's all it is the hill. To get to the top of the hill from where you start. So that would be 50. We had to add 50 kilojoules of energy to get the process started. And then let me just ask you this. Do you remember to actually start to break down glucose, how many ATP did we have to use? 2. So to even start, you know how we had to provide some energy to get glucose broken down? That energy came from ATP. So anytime we need a source of energy to make reactions or provide activation energy, we can use ATP to do that, and that can make reactions be spontaneous. Okay? Whew. I covered about a third of what I wanted to cover today. So we will continue on tomorrow. Make sure you guys watch lecture 4, and we will see you guys then.